Good morning and welcome to today's MPG Primer, and special thanks today to Scott and Russell for getting everything up and running here in the video, and to everyone who's watching either in the room or remotely for your patience as we've gotten started. We're very fortunate today to have Dr. Saori Sakaue, who's a postdoctoral fellow in the Ratchdori Lab at Harvard Medical School. In her MD and PhD at the University of Tokyo, Dr. Sakaue combined biobank scale genotype data and statistics to understand biology of complex human traits. Her current work focuses on using genetic data to distinguish between causality and consequence in human disease. In her clinical work as a rheumatologist, Saori focuses on autoimmune disease and therapeutic interventions. And we're so excited to hear about your work with HLA alleles. Just a reminder that if you are here in the room, there are or will be mics to address any or raise any questions. And then Sarah is monitoring the Q&A for those joining us remotely. So if you have questions, please post there and we will voice them as soon as we can. So thank you so much, Saori, and please get started if you'd like.
Thank you for the kind introduction and invitation to speak at this time. I'm very excited to present a statistical genetics guide to identifying HLA alleles deriving human complex diseases. So here are the outline of today's primer, here just for your reference. So first, to get everyone on the same page, I will introduce what the MHC or HLAs are, which is sometimes confusing to people, and introduce interesting biology of this region. This importance is especially highlighted in the genetics of the HLA region in complex traits, so I will explain that. Then I introduced statistical methods to impute HLA alleles from SNP array genotype data, which is now routinely used for the large-scale biobank analysis, and we can apply And using these imputed HLA alleles, I explain how we can perform association tests of these alleles to better disease. And I will briefly explain advanced methods of these HLA alleles, like non-additivity assessment and eQTL analysis. And finally, I will introduce useful resources that you can easily use for your HLA research as well. So let's get started with what the MHC region and HLA genes are in the first place. And please interrupt me if you have any questions. So MHC is an acronym for Major Histocompatibility Complex, which was first discovered in 1936 as a genetic locus responsible for tissue transplantation rejection in mice and then in humans, which later resulted in Nobel Prize in 1980s. And actually, the earliest complex human disease genetic association was reported within this MHC locus more than 50 years ago for lymphoid cancer called Hodgkin's disease. So this locus is really a historically important region for human genetics. Yes. And as time passed by, the Human Genome Sequencing Project reveals that the MHC locus consists of a cluster of more than 200 immune-related genes. So this is really a gene-dense region, including some famous genes in immunology class, such as C4a, C4b, a complement gene that can mark the infected cells, or TNF gene, a famous cytokine molecule signaling the inflammation. But among them, the most important genes for immune function are the HLA genes, or human leukocyte antigen. HLA genes are again discovered through antigenic differences between white blood cells among individuals with relationship with transplant rejection in humans. And HLA genes are broadly classified into two classes, class 1 genes, which is here on the right, HLA A, B, and C, and HLA class 2 genes, which is on the left, like these kind of genes. MHC class 1 genes on the left in this figure, like HLA A, B, and C, comprise alpha chain, which is colored in green here, of the HLA molecule, and are expressed on nearly all nucleated cells surface and present self or intracellular peptide to CD8 T cells through T cell receptor. By the way, this beta-2 macroglobulin of the HLA molecule of MHC class 1 molecule is on the different chromosome than this chromosome 6. So this is on the different chromosome of the 15. And on the other hand, MHC class 2 genes, such as HLA-DR, DP, DQ, consist both alpha and beta chain respectively, like DRA and DRB1, DPA1 and DPB1, and are expressed primarily on the antigen-presenting cells, such as B cells or dendritic cells, and present extracellular pathogens to CD4 T cells to evoke immune system and protect us against an infection. And because humans have a long history of fighting against various infectious organisms, HLA genes, especially in the antigenic peptide binding groups, like here, has extensive genetic polymorphisms to be able to present various antigens across human populations. When each individual has SNPs within the coding sequence of the HLA genes, like colored in red here, it results in variations in with amino acid sequences of the HLA molecules, especially in the peptide binding groups, like those colored in red in this figure. And based on this combination of the sequence variation in HLA, we have a name for HLA types or the alleles, like HLA-DRB1 0101 or HLA-DRB1 0401. This naming system is a little bit confusing, so I will briefly touch on this. So the HLA alleles are consisted of the HLA gene name, like HLA-DRB1, with a field separator of asterisk, and the allele names, which is again separated by colon, and it consists of 4 fields. The first field, or field 1, describes the historical broad classification based on the serology of the HLA molecule. And this field 2, or the second field, is the most important one and corresponds to the unique amino acid sequence of the gene. So all the non-synonymous changes or substitutions are reflected in this field 2. And field 3 reflects synonymous polymorphisms or substitutions, and field 4 reflects non-exonic or non-coding polymorphisms. And because these 2 field alleles are what differentiates HLA protein structure and antigen binding capacity of the peptide binding groups, it is very essential to know which 2 field HLA alleles are associated with the disease, rather than focusing on each individual SNP separately within the HLA region, just as we do in the GWAS, standard GWAS. For example, for HLA-DRB1 gene, 0401 allele is very famously known for a strong risk for rheumatoid arthritis, whereas 1501 allele is a strong risk for multiple sclerosis, both of which are representative autoimmune diseases of the joint and neurocerebral disease, respectively. So let's move on to the human genetic studies of the HLA regions and complex traits. So the MHC locus on the chromosome 6 confers the largest number of disease association of any locus genome-wide. This is a Manhattan-like plot showing how many traits in UK Biobank on the y-axis are associated with each genomic locus when we bind these whole genome SNPs into 2 megabase bin. And we apparently see an outstanding peak at the MHC locus at chromosome 6, and most of which are driven by HLA genes. In addition, MHC confers the largest amount of heritability of most autoimmune diseases of any locus genome-wide. So in this example, rheumatoid arthritis latest GWAS that we published last year, although we identified more than 100 associated loci, if you look closely into this figure, so this is the negative log 10 p-value from 0 to 1,000. And we have genome-wide significant locus more than 100. But we can clearly see that MHC locus has by far the significant effect on the risk of the rheumatoid arthritis and the largest effect size of all loci combined. But MHC is also notorious for its complicated linked disequilibrium or LD structure. There is an exceptionally long LD across the entire MHC locus, as this figure shows. So we must have a specific strategy to disentangle the MHC by fine mapping the HLA variation to understand the disease associations. And historically, there have been experimental methods to directly determine the HLA alleles, either by Sanger sequencing-based typing like this, or the sequence-specific oligonucleotide probe hybridization, or SSOP, where we specifically amplify the MHC locus by PCR reaction and determine the HLA type by hybridization with allele-specific probes. However, the important caveat for these methods, sequencing-based method, is that this is really a laborious and costly process, and it does not scale to many individuals, like tens of thousands or hundreds of thousands of individuals in the era of large-scale biobank studies. So this is why we need a method to statistically impute HLA variations from inexpensive SNP genotype data, which can be performed simply by the reanalysis of the already existing data without any additional cost. So now that I hope I convinced you that we need to impute HLA alleles, I introduce a brief overview of the statistical methods to impute HLA alleles from SNP microarray data. So as a recap for this audience, I may not have to recap this, but just to get everyone on the same page. In the commonly performed genotype imputation, we have genotyped SNPs on microarrays, which is limited in number, like less than 1 million. And then we can impute untyped whole genome SNPs by some statistical magic in order to conduct whole genome association studies, which results in tens of millions of variants in total. And in HLA imputation that I want to talk about, we have the SNP information, which is the same as the GWAS genotype imputation on microarrays, which is limited in number. But instead of whole genome SNPs, we want to impute HLA variations using the same statistical magic. So in genotype imputation, from non-genotyped SNPs, and given the reference panel, whole genome SNPs with whole genome SNPs like 1,000 genome project genotype, we can statistically infer or impute untyped whole genome SNPs to perform GWAS. But in HLA imputation, from the same non-genotyped SNPs, and given the reference panel with SNPs, HLA alleles, and amino acid sequences, the goal is to statistically impute the HLA alleles and amino acid sequences to fine map the disease-driving HLA alleles. So as a known input of target cohort, we use post-QC genotyped SNPs in the MHC region, but unknown HLA alleles, amino acid sequences, and HLA SNPs. And I'll elaborate more on how we should do the QC of the genotype in the MHC region later, because it sometimes needs specific considerations. We then utilize HLA reference panel, which includes SNPs, HLA alleles, amino acid sequences, and HLA intragenic SNPs as something known for those individuals. Given the extreme diversity across ancestries, we recently created a multi-ancestry HLA imputation reference panel covering major populations to enable accurate HLA imputation in global population. And also I will touch on this later in this session. We then statistically phase haplotypes of the target genotype and impute the untyped HLA alleles, amino acid sequences, and SNPs, which were missing in the input, but now we can infer this from this statistical imputation. And as a phasing and imputation engine, we can basically use the same algorithms we use for genotyping imputation. The famous softwares including like BEAGLE, SHAPEIT in EAGLE for haplotype phasing, and imputation uses BEAGLE and Minimac, which can be just used as an application, further application from the genotype imputation. And in this framework, our group has previously developed this SNP to HLA software as one of the most popular HLA imputation software using BEAGLE as an imputation engine. But we now implemented this using EAGLE in Minimac 4, which is a lot faster than BEAGLE and computationally scalable to hundreds of thousands of individuals, like biobank scale data. Also, this implementation is freely available at Michigan Imputation Server, which is a publicly available web-based imputation server hosted by Michigan University and also our group, with the availability of the latest multi-ancestry HLA imputation panel. So I will also touch on this later in this session. So now I explain the HLA imputation step by step. If you have genotype data of standard SNP array of your cohort, you do the genotype QC like standard one that you usually do for any GBAS. So you start with per individual QC and per variant QC, just like the standard GBAS you do. But there are also specific considerations for the HLA region. For example, when you have genotype data from your cohort and you want to compare the allele frequency of your genotype data against the allele frequency of the reference panel as a QC procedure, we should note that allele frequency within MHC are inherently very different across populations. So if your cohort does not exactly match the population in the reference panel, then it might be somewhat expected that allele frequency does not perfectly align, but it is permitted for the MHC region. Similarly, in the standard GBAS QC, we use the deviation from the Hardy-Weinberg equilibrium as a metric for genotyping error, as shown in this bottom plot. So in this figure, the top one is showing the genotype clustering with correct genotyping, where we can correctly assign the genotype 0, 1, 2 by using this plot. But when the genotyping was difficult, there is difficulty in distinguishing these clusters separately. So this causes wrong assignment of the genotype. And this can be detected by analyzing the Hardy-Weinberg equilibrium. However, because MHC is in the strong natural selection, it is to some extent expected that MHC variants deviate from Hardy-Weinberg equilibrium. So we may consider more lenient threshold for doing HWE-based QC than we do in the standard GBAS. So these are specific considerations when QCing the MHC region, so that we don't lose any important markers from your target genotype data. So once we get post-QC data, if the genotype data can be uploaded to the public server, you can use the Michigan Imputation server for HLA imputation. And if it's not allowed to upload that to public server, you can also use the in-house imputation in your server, depending on the sample size. So the basic SNP to HLA imputation is suitable for imputation in smaller cohorts because BEAGLE is less computationally scalable. And we might consider imputation with EAGLE for phasing and Minimac for imputation for more larger cohorts like biobank scale data, like tens or thousands, hundreds of thousands of individuals. And I finally briefly explain how to interpret and use the HLA imputation results. The HLA alleles and amino acid sequences in the imputation reference panel, as well as post-imputation file, are coded as binary markers, where the reference allele A indicates the absence of the allele, and the alternative allele T indicates the presence of the allele. So in this example, this one, haplotype A of one individual, if we have DRB101 allele for this individual, we should have one coding for the DRB101 allele, which is designated by the alternative allele of T, but 0 for the other alleles, because this is not 0401 or 0801. And because two-field allele can determine the amino acid sequences of this HLA molecule, HLA DRB1, we can naturally or obviously assign amino acid sequencing just based on this two-field alleles information. For example, for this position, negative 25, DRB101 should have K at this position, but not R for this position. Then we should assign 1 for genotype for negative 25K, but 0 for negative 25R. In the same way, at negative 24, O4 you have L letter or amino acid sequence for this position, but not F. Then we can assign 1 for negative 24L, but 0 for negative 24F. Accordingly, the post-imputation VCF file looks like this. This is exactly the same format of the standard VCF file for genotype imputation. So the first column here represents the chromosome number, which is always 6 for the MHC region. And the second column indicates the genomic position of this variant. And the third column is the name of the imputed alleles, and with reference allele A, which indicates the absence of the allele, and the alternative allele T, which indicates the presence of the allele. For example, for this allele, HLA-A0201 allele, we can see the info score at this field. So the imputation R-square here is good at 0.988. And this first column indicates the genotype of the first individual, which is estimated to have just one copy of this HLA-A0201 allele, because there is 0 bar 1 genotype here. And on the right side of the column, we have this number 0.996, which is the dosage of this allele, which indicates how many copies of alleles this individual has with uncertainty taken into account. So this is most likely to be one copy, but there's less probability of just having one copy. So this is why we have a float number at this dosage position. And we already confirmed the high accuracy of the HLA imputation without using 1,000 genome project samples across all ancestries. So now that I talked about HLA imputation, I would like to switch gear to move on to the methods to perform association tests using the imputed HLA variations. So the basic concept of the association tests are again the same as the standard GWAS. So I'll first introduce how to do the simplest single marker test to assess the association between the single HLA allele with the disease like I show in this figure 1. And the next HLA specific analysis is an Omnibus test, which is in this second part of this figure, where we want to determine which amino acid position within HLA molecule is driving the signal. Here, we have to assess multiple patterns of amino acid residues beyond 2, like multi-allelic SNPs in the GWAS. And I finally introduced the third analysis, which is conditional haplotype test, which is somewhat similar to the conditional analysis in GWAS, where we want to assess the combination of independent amino acid positions that together explain the disease associations. So the first association test, a single marker test, is to ask which single HLA allele affects the disease risk in a conventional logistic regression framework. And this is pretty similar to what we do in GWAS, so asking SNPs association with the disease risk one by one. So here we associate the binary coded disease phenotype for individuals, which I colored in yellow here, and associate that disease phenotype with an imputed allele dosage of a single HLA allele, one at a time, which is shown in blue here, with accounting for GWAS covariates such as age, sex, and genotype pieces, which I show here in green. So this is really exactly the same framework that we do in the GWAS SNP association tests. The Omnibus test is to ask which HLA amino acid position within HLA molecule affects the risk of the disease. So as I explained, each amino acid position has multiple possible amino acid residues among individuals within HLA molecules. For example, in the HLA-DRB1 gene, in this position, position 11's amino acid residues, there are 6 possible residues of amino acid. So here I show the example amino acid sequences, and the position 11 is colored by various colors. And we have potentially 6 possible amino acid combinations like L, S, V, G, and D, and P. We can encode these individual amino acid residues from imputed amino acid dosage from the HLA imputation, like this matrix here, where each column represents the residue, amino acid residue, like L, S, V, G, D, and P, and each row represents an individual. So each row should sum up to 2 because we have 2 copies of DNA. Then we can feed this matrix how each individual has amino acid residues for this position 11 for the HLA-DRB gene in this logistic regression model with dropping the final column P because it can be obviously determined by the remaining 5 columns by summing up these 5 columns and then subtracting from 2. We then compare this full model with including the amino acid variation for this position against the reduced model only accounting for the covariates, but without including the amino acid information. And by performing the ANOVA test or F test, we can ask how much this amino acid position's genetic polymorphism increases the explained variance for the trait of your interest and determine the single most significant amino acid position. I hope this is clear to you, and as an extension of this Omnibus test, the conditional haplotype test asks which HLA amino acid position in combination affects the risk of a disease. So the goal is to determine the independent amino acid sequence positions within HLA molecules as a haplotype that influences the trait variance most. So this is very similar to the conditional tests we do in GWAS, but we also have to account for the haplotype information for the HLA and also the multi-allelic nature of the amino acid residues rather than having just 2 types of the alleles in case of the SNPs in GWAS. So as a recap, the Omnibus test was able to determine the single most significant amino acid position for the disease. In this example, position 11, and we had 6 possible amino acid residue variations. We want to next ask which additional amino acid position as a haplotype most explain the trait variance. To do so, we utilize the 2-field HLA allelic imputation results because it can account for haplotype information. So each 2-field HLA alleles is a unique sequence of amino acid residues like this. Therefore, when we think only about this position 11, the 2-field HLA alleles can be naturally categorized into 6 groups each of which corresponds to 6 unique possible amino acid residues. So like in this bottom figure, the L is consisted of 2 field alleles O1, O1, or O1, O2. S is consisted of O3, O1, O8, O1, and so on and so forth. And next, we want to assess the second position. In this example, the position 71, here colored in green and red and blue. We can now group 2 field HLA alleles into 10 groups if we consider both position 11 and position 71 simultaneously, like L plus R or S plus K or V plus R. So if we exhaustively count the number of combinations at 11 and 70, there are 10 groups like in this right bottom plot. And then we can compare the or we can calculate the explained variance by these 10 groups of variations of amino acid residues from positions 11 and 71 and compare that variance against those with a variance explained by 6 groups, which only account for position 11.
There was a question in the Q&A, I think, just relating back to the VCF you made with the imputation and how all of the alleles in that VCF were A and T, but now you have many different alleles, and just how it is you went back from those kind of from this per variant VCF to now going back to the actual amino acids.
So usually we have to have some script to convert that information. So because we should have 2 alleles for each gene, we have to guess which is the most possible 2 HLA alleles for each gene from the dosage information of each one of the HLA genes. That's a good point. We have to convert that information to obtain the actual amino acid sequences for the imputation.
The T is like a placeholder that you then loop back into the actual sequence. Thank you.
So let's go back to the conditional haplotype tests. So I think I believe I explained the setup, and now I will write this into the mathematics formula. So the full model will be including the 10 groups from the combinations of the positions 11 and 71. In this example, there were 10 possible groups. And the reduced model includes 6 groups only from the position 11. In this example, there are 6 groups. We then compare the explained variance between the 2 models or difference between the explained variance between the 2 models by using the ANOVA or F test until we exhaust all independently associated positions. This sounded a little bit complex, but actually this conditional haplotype analysis was really important and effective in defining precise biology. In this 2012 publication by my PI, Richard Lee et al., the significant association with rheumatoid arthritis risk was mapped to the positions 11, 13, 71, and 74 of the HLA GRB1 molecule, which coincide with an antigen peptide binding group of the HLA molecule, which is very biologically reasonable because if we have amino acid variations for the antigen presenting site, then maybe autoantigen, our capability of presenting autoantigen might differ across individuals, which could contribute to the risk of developing autoimmune diseases. So let's move on to 2 additional advanced methods in the HLA. The first one is to test the non-additivity of the association. So all the association tests that I've explained so far was assuming the additive model, which is on the left, where each 1 copy of disease-associated allele contributes to the disease risk with the same amount. So we assume the linear relationship between the dosage of the disease-associated allele and log-odds ratio. However, if we think about the biology of the HLA, having just 1 allele or 1 type of the HLA protein structure on the cell surface might be enough to cause a risk of the disease, like autoimmune diseases. In that case, it will follow the non-additive model on the right, or a dominant model, where there is a deviation from the linear relationship between the dosage of the disease-associated allele and locus ratio, and there is additional contribution D at the heterozygous genotype. In assessing this non-additive effect, we can both include the conventional additive term X, which is just the number of alleles of the disease-associated allele that we have been using. And also, we can simultaneously include the non-additive term delta, which is 1 if and only if the genotype is 1 or heterozygous. Like in this example, the individual 2 and 4 have a dosage of 1, so they have a delta of 1. But individual 1 and 3 have a genotype of 0 and 2, so they will have a non-additive term of 0. And by using this analysis, we can both assess the additive effect and non-additive contribution for the disease risk for each single HLA allele associations. And we, in fact, revealed that the HLA associations in autoimmune diseases such as psoriasis, type 1 diabetes, and rheumatoid arthritis had a strong non-additive or dominant effect, which is biologically, I think, interesting and insightful. The final method is the expression quantitative locus or eQTL study in the HLA genes. So while we thought a lot about the coding variation within HLA so far, in the eQTL analysis, we assessed how the cis-regulatory SNPs nearby the HLA genes affect the level of expression of the HLA genes from the RNA sequencing data. So in this example, the cis SNP, whether we have cis SNP of green or red, affects the gene expression abundance of the HLA DQA1 gene. The eQTL is now a standard method, but there is also a specific challenge in the eQTL study for this particular HLA region. After we do standard RNA sequencing experiments, we typically align the reads to the reference genome to quantify the expression abundance of the genes by counting the number of reads that mapped to that particular region. However, in the case of the HLA genes, if we only use a single human reference genome, like GRCH38 from just one representative European individual, there will be a mismapping of the reads because the region is too highly polymorphic, or the multi-mapping of the reads because of the similarity among the different HLA genes. And this mismapping and multi-mapping is in fact a critical issue in the HLA region and could lead to an inaccurate quantification of the HLA expression, which is a critical issue when we think about the eQTL study. We have to associate the genotype and the HLA expression abundance, the true biological expression abundance. So to address this issue, Joyce from our group has led a study for accurate eQTL mapping in single-cell RNA-seq data. And the key idea is that instead of using a single human reference genome, we mask the reference for the HLA gene site by N. And instead of using that single reference genome, we use the personalized HLA alleles as additional mapping reference that we can obtain from the statistical imputation of the HLA alleles from the genotype data of the target cohorts. We show that this personalized reference mapping strategy from the RNA sequencing data resulted in by far more accurate quantification of the HLA genes in multiple data sets that resulted in more accurate identification of eQTLs in the HLA region. So finally, I introduce useful web resources that you can use for the imputation and association tests. So as I explained, the Michigan imputation server implements all necessary steps for the HLA imputation, including the availability of the latest multi-ancestry HLA reference model. So you can freely sign up for an account by visiting this web page, and you can Google just Michigan imputation server. Please click on the Run tab and choose the job for the genotype imputation HLA with Minimac 4. you can select the latest HLA reference panel by clicking the 4-digit multi-ethnic HLA reference panel version 2. Then all you have to do is to just upload the post-QC genotype of your cohort on chromosome 6 in the VCF format. You will be notified once the imputation is completed. We also host a tutorial website at our GitHub, here, with detailed command line scripts, including the genotype QC, phasing in imputation and association test, which is basically all the steps that I talked today. So if you are interested in doing the analysis, HLA analysis, you can refer to this website just for your reference. So to summarize, I think I have convinced you that HLA genes are one of the most important and polymorphic regions among the entire genome, and we can statistically infer individual HLA alleles from SNP genotype data by using statistical magic or imputation. And there are several specific methods like Omnibus test or haplotype, conditional haplotype test used for the association studies for the HLA alleles in disease of interest. And these kind of methods are summarized in our manuscript, which is now available at the bioRxiv and soon in press at Nature Protocol. So if you also are interested in the methods or mathematics behind my talk, you can visit the bioRxiv website. And with this, I would like to acknowledge all the collaborators in our lab and also outside of the lab, especially Shomo, my PI, and Paul, who originally developed and snipped the HLA imputation framework, and also people in the Michigan imputation server who kindly implemented our strategy at their imputation server. And I'm happy to take any questions.
A couple of questions online, but if anyone in the room has questions too, feel free to grab a mic. One of the questions was whether in your models, when determining between additive and non-additive, are those mutually exclusive categories, or is there any kind of in between about them?
Yeah, so that's a great question. So when we think about the non-additivity, we can quantify how much this dominance or non-additivity exists by quantifying the effect size of D. So in a very extreme case, A equals to D, where there is a complete dominance inheritance model. But if D is a little bit less than A, then it's incomplete combination of the additive model and non-additive model. It can also be rather negative D. In that case, it will follow the recessive model. So we can determine how much the dominance term exists by quantifying this D from the data.
Thank you. Another question from the Q&A was whether the TopMed data for imputed HLA genotypes is available.
Unfortunately, it cannot be accessed freely or publicly from for the raw genotype and raw sequencing data for the TopMed. So if you want to, if you are interested in doing HLA imputation using the TopMed reference panel, you have to upload your genotype at the Michigan imputation server where the raw genotype is securely stored.
That makes sense. One of the things you mentioned at the beginning was the necessity. It seems like there's two opposing statistical problems. One is which you need your reference panel to well match your study panel, but then you also need to account for novel sequences within the HLA locus. So how do you balance those two things? Like what statistical choices did you make?
Yeah, so you're right that people are continuously reporting novel HLA alleles from sequencing data. In that case, we cannot account for that novel allele in the HLA imputation because we don't have that allelic information and haplotype information for our HLA imputation panel. So if you really suspect there may be a contribution from the novel allele, then what you want to do is to sequence, Sanger sequence or target sequence your MHC region and then define disease, novel disease-associated alleles. But for almost all the commonly observed HLA allele, the HLA imputation can confidently cover for the accurate imputation. So in that case, you don't have to do a sequencing study.
That makes sense, common versus rare approaches.
That was a super interesting talk. Thank you so much. I'm curious about when you were talking about sort of determining the number of amino acids that help to better explain whether a disease is present or not, whether you have, from looking at various diseases, have a sense of how quickly that falls off. That is it likely to be the most important amino acid accounts for a lot of the variance, and then you get a steep fall off, or is there some pattern across different disease that differs across different disease types? Or
so in the case of the rheumatoid arthritis, we have so in this 2012 publication, identified 4 possible independent amino acids. But in the later largest study that we conducted last year, we defined 3 additional amino acids just due to this. We have much larger sample size and microarray data, and just statistical power increased. And if we do other disease analysis, we get similar combination of the amino acids like 71. We frequently observe 11 in position 71, but the remaining amino acids like 74 yet to be found. So maybe it's a trade-off between the statistical power and how important these amino acids are for the presenting capacity of their autoantigens.
Question is, this has obviously been very high yield when studying disorders that are more obviously related to immune dysregulation. Do you have like the next tranche of like disorders where you think that there might be maybe a smaller magnitude of contribution to the HLA locus to disease, where we're finally getting to enough people to detect it?
Yeah, so some people say that all diseases are related to immunology. So in that sense, maybe, that's why maybe why we have this many number of associations in the UK Biobank, like more than 70 diseases associated with MHC locus. So we have a very strong association for cardiovascular disease or neuropsychiatric disease like schizophrenia. And in the case of the schizophrenia, actually the driving molecule in the MHC region was not solely by the MHC, but it was driven by the complement genes like C4, which is, I think, reviewed by Steve McCarroll's group a few years ago. So maybe if we go deep into cardiovascular disease, there are maybe different mechanisms that we are seeing for autoimmune diseases. So I think I agree that it is the next challenge that may be interesting.
Thank you so much for an excellent talk this morning, and hope you have a wonderful day.